life cycle of Fasciola, that is Liverflukes, Fasciola, Fasciola hepatica, Fasciola gigantica. So when we talk about the Liverflukes, it is under the trematode, and these Liverflukes are leaf-like or leaf shape in appearance, and these Liverflukes are diagenine. They have two hosts. We have the primary host as well as the secondary, and then the intermediate host, as well as the secondary or the intermediate host. So when we talk about the primary host, we can have a farm animal like the sheep or cattle, and the secondary host is a water snail. So the organism completes its life cycle in two organisms. It is a parasite. We all know that it is an endoparasite, and it has different stages: the egg stage, the larva stage, as well as the adult stage. So we are going to understand the life cycle of this organism. First of all, this organism is found in the liver. That's what we call it Liverflukes, in the liver of a farm animal. Let's say the liver of the adult Liverfluke, in the bioduct. This adult fluke in the bioduct carry out cross fertilization, and after cross fertilization, you can also carry out self or cross fertilization inside the bioduct. And after cross fertilization has taken place, then we can now have the fertilized egg. The fertilized egg now will now pass out. After they have carried out fertilization in the bioduct, the fertilized egg will pass out through the bioduct to the gut, and then it pass out out of the body as feces, just like when normal sheep remove feces or cow. So the eggs that are already fertilized passed out of the body or through the gut, and exit as feces. So this egg will move out as if the host, that is the sheep or the cattle, that is the host. So it will pass out with their feces. So these feces, the eggs now, there will be a larva. It will develop into the first larva called myocardium. Myocardium will age out. Mycelium age out of the egg. So the egg will age out as myocardium, and this myocardium is the first stage, the first larva stage. That is myocardium. And this myocardium is ciliated. It has numerous cilia for locomotion, and also the mycelium have structures for locomotion. We said they are ciliated, and the main work of the mycelium is to locate the secondary host, because it just leaves the primary host. So the mycelium will not take longer. It will just find way to infect a secondary host. So it will swim and infect a secondary host. It will swim, the mycelium will swim and penetrate a secondary host. a water snail. A water snail. That is the secondary host. So after the miracidium swim, after the penetration has taken place, okay, the miracidium will develop into a sporocyst. And this development is as a result of the fact that the cilia will be withdrawn, the outer cilia will be withdrawn. So the miracidium will develop into a sporocyst. Miracidium will develop into a sporocyst. And the sporocyst is what? The second larval stage. Sporocyst is second larval stage. Larval stage. And this sporocyst also will carry out development, and it will later develop into the what we call radia. The sporocyst develop into the radia. Sporocyst develop into radia, and this radia is called the third larval stage. Radia is third larval stage. So those are the larval stage of this organism. So from the radia, you now have the saccharia. So this saccharia can develop from the radia, and the radia can also multiply, because the radia is responsible for multiplication as well as feeding, because the radia has developed some structures that enhance it to feed, just like a development of a muscular pharynx that enhances to acquire food. Then this radia will migrate into the intestine, the digestive system of the secondary host. We are still inside the digestive system of the host, which is the water snail. As from the miracidium, it developed into the what's it called? The sporocyst. From the sporocyst, it developed into the radia, still inside the digestive system. So it moved to the intestine, the digestive tract of the snail, where the radia will develop into the saccharia. The radia multiply to form more radia or saccharia, depending on the condition of the environment. So the saccharia has developed a lot of structures that make it resemble the adult liverfluke. So this saccharia, the next thing is that the saccharia will insist. The saccharia will insist and to be inside water or on damp vegetation. So the saccharia has already developed a lot of structures. The saccharia have the oral and ventral sucker for attachment. The saccharia also has tail, which enhances the locomotion. So the saccharia has gut, it has a system. So the saccharia now develop into a cyst, like it insists. It has a cyst wall, it has a cyst producing cell. So this cyst producing cell we have insist. So the saccharia insists. Saccharia insists on damp vegetation. So the saccharia are picked up by farm animals when they feed on vegetation. The saccharia are picked up by farm animal. It is ingested by cattle, or let's say by sheep, or during grazing. So when the sheep are there to feed on grasses, the cysts of the saccharia are being ingested by the cyst. So this saccharia will insist when it gets to the digestive system of the sheep, and cyst, alright, or saccharia emerge and develop into a young saccharia emerge and develop into a young fluke that is inside the alimentary canal. So this can now move to the liver, from the liver to the bile duct. So that is how they carry out their life cycle. So from the primary host, this is the sheep, the liver, the liver saccharide emerge and develop into a young fluke in the liver. So from the liver, the adult fluke will emerge in the bile duct and the carry out self-transmission. Just the way that is how the life cycle looks. So we also have their effects. When you talk about liver fluke, they have a lot of effects, parasitic effects on the human as well as the environment. First of all, they cause disease, and this disease lead to a lot of death. When they have high infection, heavy infection, it can lead to death. Those are the parasitic effects. And they can also cause liver rot, which can lead to dropsy. Those are the parasitic effects. And also the waste product, the excretory product of this parasite is very toxic to the primary host. So it can lead to disease, liver rot. I talk about liver rot, it can lead to dropsy. So those are the effects. Then we also have the control. When we want to control this Fasciola, we can actually introduce biological control to introduce duck or geese that feed on this nail. Then we have to grade, we have to remove, we have to clear their habitat. We have to clear the pasture or the land where they are found. You can also kill the larval stage. You can also kill the larval stage with some chemicals. So those are the controls. Those are the controls of liver fluke. So that's all about liver fluke.